two kinds of nucleic acids, RNA and DNA. DNA or deoxyribonucleic acids is an incredible molecule that carries genetic information for the development and functioning of an organism. They are located within the nucleus as a chromatin. During the time of cell division, this chromatin structure forms the chromosomes. So the basic structure of chromosomes are nothing but DNA. So now we would study the structure of DNA. In 1953, James T. Watson and Francis Crick proposed that DNA has a double helix structure on the basis of X-ray diffraction theory, and in the year of 1962 they have received the Nobel Prize for their work. The double helical DNA is a polymer of a macromolecule known as nucleotide, and it looks like a twisted ladder. Nucleotides make the strands of double-stranded DNA. So now we would understand the structure of nucleotides. Nucleotides are composed of three main parts: a nitrogenous base, a pentose sugar and a phosphate. First, a base and a sugar attach to each other. These forms the nucleoside, and when a phosphate binds to this nucleoside, it forms the final structure which is known as nucleotide. Now we would understand the structure and types of nitrogenous base, the first part of nucleotide. Nitrogenous bases are of two types, purines and pyrimidines. Then the purines could be divided in two types, adenine and guanine. Both the purine contain two rings, one is hexagonal and the other one is pentagonal. Pyrimidines are of three types, cytosine, thymine and uracils. Among them, the cytosine is present in all kinds of nucleotides, but the thymine is present only in DNA and the uracils are present only in RNAs. Like the purine, the pyrimidine contains only a single ring structure. Now we would understand the structure of pentose sugar or the 5-carbonated sugar, the second part of nucleotide. The pentose sugar contain a 5-carbonated ring or a pentameric ring structure. There are two types of pentose sugar, ribose and deoxyribose. Now students, try to find out the difference between them. Yes, the missing OH with the second carbon. Ribose are present in RNAs, while DNA contain the deoxyribose pentose sugar. So thus these nucleotides forms the strands of DNA. The sugar and the phosphate forms the backbone of the structure, and the nitrogenous base makes hydrogen bonds to bind them together. In this structure, adenine always binds with the thymine, while the guanine always binds with the cytosine. Adenine and thymine forms two hydrogen bonds, while guanine and cytosine forms three hydrogen bonds. Both the strands wind around each other and forms this double helical twisted ladder-like structure. One complete turn of this structure forms a major groove and a minor groove. One complete turn contains 10 base pairs and it is 34 angstrom long. That means the distance between two base pairs are 3.4 angstrom. DNAs have some incredible properties as they can make their own copies. Now we know that DNAs are made up of nucleotides or nucleic acids. These nucleotides or the nucleic acid has a property to replicate or make its own copy. That's why DNA can also make its own copy. And thus, DNA makes the molecular basis of our inheritance. They can store molecular informations in their specific regions known as genes. So, genes are segments of DNA which can control the physical characteristics of individuals. So, these genes are present on DNA, which forms the chromosomes during the cell division, and thus they got transmitted from one cell to its daughter cell. That's why DNAs are known as the molecular basis of inheritance. So, students, do you want to know about the other nucleic acid or RNA? Then subscribe the channel and press the bell icon to get the notification of the next video. Till then, bye bye.